This is the structure of Alectinib. Alectinib is used in the treatment of anaplastic lymphoma kinase positive lung cancer, as well as metastatic non-small cell lung cancer. Alectinib is an inhibitor of the anaplastic lymphoma kinase, or ALK tyrosine kinase receptor. ALK plays a role in normal human nervous system development, but genetic mutations and alterations can lead to overexpression of ALK in tumor cells. So Alectinib was developed through a high throughput screening program in Japan, brought to market in 2014 in Japan and in 2015 in the US. And it was designed to overcome drug resistance that had been developed to first generation ALK inhibitor crizotinib. So if we look at the molecule here, we'll see the morpholine ring at this end with an oxygen and a nitrogen, and a pyridine group here with a nitrogen, and then a benzene ring with a carbon substituent here, and a 6-member carbon ring with a dimethyl group and a carbonyl group, and then a pyrrole here with a nitrogen and a benzene ring, and finally at this end a nitrile group here. So it is a lipophilic drug, and it binds at the ATP binding site of the ALK receptor. And the first thing we'll notice when we look at it bound to the receptor is that this portion of the drug is quite exposed, so the morpholine and pyridine end of the drug, and this was designed during optimization to enhance the pharmacokinetic properties of the drug, so to increase solubility and bioavailability of the drug. We do see a strong hydrogen bond here between the carbonyl group of the drug and this methionine residue up here. And it's not shown here, but there is a hydrogen bond between this nitrile group and this nearby lysine residue as well. And there are many hydrophobic forces interacting between the hydrophobic portions of the drug and in particular the many leucine residues surrounding the drug. And there's one leucine residue in particular worth mentioning, so this one here, and this residue is the site of mutations that confer drug resistance to crizotinib. As I mentioned, Alectinib is a second-generation ALK inhibitor, and it retains its potency despite the presence of mutations at this leucine residue or other amino acids as well. So it is a very selective and highly potent ALK inhibitor, and is very efficient at inhibiting this receptor, even in the presence of drug-resistant mutations.